So good afternoon all of you. You're able to see the screen? Okay. So today we are going to see about gel filtration chromatography. So first we are going to see about gel filtration chromatography followed by SDS page. Okay, so these are two very important techniques in chromatography as well as in electrophoresis for the separation of proteins. Okay, so chromatography you all know, as you can see in the slide, it is a technique used for the separation of chemical substances and also biological compounds, okay, and particles by how they are separated. The substance or the sample to be separated are allowed to move through or along two different phases. That is the stationary phase and the mobile phase. So stationary phase is made as stationary. Okay, it is made to stand. Either it can be a solid or it can be a semi-solid. Okay, or also a liquid, but it cannot be a gas. Whereas the mobile phase can be a liquid or a gas, but it can never be a solid. So that it can move or move through or move past the stationary phase. So today what we are going to see is about the gel filtration chromatography. So gel filtration chromatography, as the name implies, you can understand that it is a gel. Okay, what is a gel? You would have come across a lot of gel. Okay, in the food what you eat, you can understand what is a gel. It is a semi-solid, soft. You would have eaten jelly or gel-like food substances. Okay, it is very soft. Sometimes, no, certain food, if you put add some water for example, the resin. Okay, resin, the dry resin, grapes. When you put it in water, it can absorb water and it can swell. How does it swell? There are some pores on the surface of the resin. Okay, through the pores, water is absorbed or water enters into the resin. Okay, it acts as a semi-permeable membrane. Okay, just understand that it has many pores, and through the pores, the water enters and it swells. That is one example. Okay, another example I can say, I don't remember the name, gel jeera or something, no, which you add in ice cream and other things, no. That is a jeeragam like kaduk like substance, which you put it in water. Do you remember the name? Can anyone tell me the name? Jal jeera. Pardon? Yes, can you repeat? Sabja. Yes, exactly. Tell a little louder, okay? Sabja weather, sabja seeds. Okay, that will also, if you soak it in water, it will swell. So, how do they swell? They have some pores on the surface. Okay, through the pores, water enters. Okay, and it swells. So, this is the kind of thing. And gel, no, jelly. You jelly, sometimes you need to heat it up. Okay, so what happens? It absorbs water and swells. The same kind of mechanism you have to understand, okay, about the gel filtration chromatography. The gels or the stationary phase which are used in this particular chromatography has a nature where when it is mixed with a suitable solvent, it can swell up in size. Okay, that is water enters. How does they swell? How do they swell? Okay, it has some pores on the surface and water enters through the pores. Okay, and it swells. So it inside the gel, it has a certain amount of water content, and the pores are also it is visible, so that not only water, some other molecules which are having smaller in size, okay, which are smaller in size, that can enter the pores of the gel, can also enter along with the water and can get trapped inside the pore. So this is very important phenomena which you have to understand. Okay, so this is one thing, that is each and every individual I will explain to you very clearly what is the principle behind gel filtration chromatography, and also show you some animation so that you can understand in a very well manner. Okay, so here, this is the gel, this is the property of the gel filtration. So it acts as like a filter. You have already have explained to you what is the role of a filter or a sieve. Okay, the filter has many small pores. So the pore size of the filter may vary. Accordingly, it can filter smaller particles or bigger particles according to the size. So this is very important. So it acts as a gel so that it can trap or entrap. Okay, so gel permeation, the other names given for gel filtration chromatography is gel permeation chromatography. Permeation means it permeates certain, not only the water, certain molecules. Here, for example, protein molecules. So we are going to separate. What is the objective? We are going to separate a mixture of proteins based upon the size. Some proteins will be larger in size, some protein will be very small in size, and some protein will have the size in between these two. Medium or intermediate size small protein molecules will be there. So with all this proteins in the mixture, we have to separate them based upon the size. Okay, so over here the larger molecules will come out of the column or will elute out of the column first, followed by medium sized or intermediate protein molecules, and then the smallest protein molecules which are trapped inside the gel. Okay, so you have to be very, very clear. So what is the gel? Gel is the stationary phase. It can be Cephadx, okay, it can be silica gel, which are able to swell, which have the property to swell when it is mixed with a suitable solvent. So this is otherwise called as molecular exclusion chromatography. That means the gel will permeate or allow the smaller molecules to enter the pores and get trapped into the gel, whereas the other bigger molecules are excluded out of the gel. Okay, they are not allowed to permeate or enter the gel pores of the gel. So this is the other name given. So what are the names given to the gel, this particular type of chromatography? Gel filtration, it acts as a filter. The gel acts as a filter or a sieve. Okay, so gel permeation chromatography is another name which is given to this type of chromatography, where it allows or permeates some protein molecules which are smaller in size and which can enter the pores of the gel and get entrapped into the gel. The other name is molecular exclusion chromatography. The bigger molecules of proteins which cannot enter the pores of the gel, okay, will be excluded from the gel. And where will they go? They will remain in the mobile phase, which are present in the column, okay, in the stationary phase in between the gels. So you can imagine the gels as particles, okay, small particles. Okay, gel, say you can say, or 10 particles or 100 particles. So I always told you, okay, there are pores. Okay, there are pores means here the intermolecular spaces in the stationary phase. So the intermolecular spaces or the pores of the column bed, you call it as the column bed, can either be small in size or bigger in size. How the size can be altered? The size can be altered by changing the concentration of the gel. For example, Cephadex. If you increase the concentration, the number of gels will be more and the intermolecular pore size spaces will be lesser. Okay, when the concentration is less, then the intermolecular pore size spaces inside the column will be more. So when the pores are smaller, it will allow only small molecules to pass through, whereas when the pores are bigger, it will allow the bigger molecules to pass through the gel. So this is very, very important. I hope you are able to see the screen, right? Somebody entered now. Okay. So this is about the gel filtration chromatography. So here you can see the thing is, the principle behind the gel filtration chromatography is, you can say it is exclusion, you can say it is permeation, and you can also say it is filtration. So this is what it is. The mobile phase involved is a liquid. So the liquid has to pass through the column. So when you take the column, when it is packed with the stationary phase, it should not be packed as a dry stationary phase. The stationary phase should always be mixed, okay, pre-mixed with a suitable buffer or a solvent, and then allowed to pour into the stationary into the column. Okay, so the column, the solvent is mostly used is the mobile phase, okay, or the solvent. Okay, so what happens when the mobile phase flows through the column through the stationary phase? That is the mobile phase can flow in between the intermolecular spaces of the stationary phase. So this is called as the void volume. The mobile phase which is flowing through the column through the stationary phase where it is flowing in the intermolecular spaces of the gel, it is called as the void volume. Okay, here some of the mobile phase can also enter the gel and pass through the gel, and they may also come out. So this will allow the entrapment of the molecules in the beginning of the column chromatography as well as at the end of the column chromatography. It can also help in the removal or exclusion of those small sized protein molecules from the gel to be eluted out in the end. Okay, so this mobile phase which is trapped inside the gel, it is called as the mobile phase which is present inside, okay, inside the gel. And the total mobile phase which is present inside the column, that is the mobile phase trapped inside the gel and the mobile phase which are present in the intermolecular spaces, together it is called as the total mobile phase. So this is, you can say, when you take the stationary phase inside the column, it is the bed. Okay, column bed, you call it as the column bed. The stationary phase packed inside the column is called as the column bed, and the mobile phase flowing into the through into or through the stationary phase. Okay, it is the, if it is a liquid, it should be the total volume of mobile phase present inside the gel and the volume which is present outside the gel, that is in between the intermolecular spaces, that is called as the void volume. When you add together, you get the total volume of the mobile phase. Okay. Now, what is the principle? It separates molecule. I told you the separation depends upon the difference in the size. Okay, that is the main principle in gel filtration chromatography. So here the separation separates molecules according to the differences in the sizes as they pass through a gel filtration medium packed in a column. As I already explained to you, I hope it is very clear, isn't it? So how the protein molecules are getting separated. Smaller protein molecules get entrapped in the gel, whereas the bigger molecules which cannot enter the pores of the gel may not get entrapped in the gel but will be present in the void spaces, intermolecular spaces between the gel in the column. So when the mobile phase is flowing through the column or the stationary phase, the bigger, larger molecules which are not entrapped in the gel will be washed out of the column first. That means it will be eluted out of the column first. So this is the principle. Okay. The other one is that the smaller molecules which are entrapped in the gel will be eluted at the end. Okay, in the end. In between, there are some molecules which will be, the size will be in between, the smaller and the bigger. So they will be eluted after the bigger sized molecule. Okay, so unlike ion exchange or affinity chromatography, molecules do not bind to the chromatography medium. So in the ion exchange chromatography, you have seen that the stationary phase will be either positively charged or negatively charged. So the sample of opposite charge will come and get attached electrostatically to the column. And also in the affinity chromatography, what happens? The molecules, the biological molecules, which can be a protein or a nucleic acid, which have to be separated, they will be charged. Okay, and also they have some affinity. Okay, some unique affinity to the ligand, which are present in the stationary phase. So they bind with a unique affinity to the stationary phase. So unlike that, there is no binding or affinity of any particular molecules to be separated to the column in the gel filtration chromatography. So it is used to separate biological molecules, and also determine after separation you can determine the molecular weight. After separation you can determine the molecular weight and also the distribution of the polymers. That is very, very important. This particular gel filtration chromatography not only separates the proteins based upon the difference in size, but also it is used to estimate the molecular weight of the protein. Okay, now here you can see the technique can be applied in two distinct ways. One is group separation, another one is high resolution fractionation. So in group separation, components of a sample are separated into two major groups. That is two groups. One is according to the size, bigger size, and the other one is the smaller size. So small molecules such as excess salt or free radicals are easily separated. Okay, desalting. So desalting. So this is a desalting is a method which is used to separate the proteins from salts. Okay, so this is a method, group separation method. Another one is high resolution fractionation of biomolecules. So components of a sample are separated according to the differences in their molecular size. That means not only two molecules, the range will be in between. It can separate molecules of larger size, smaller size, and also molecules of intermediate sizes. Okay, so high resolution. So what is partition coefficient? So the basic principle here is partition coefficient. What do you mean by partition? Partition or distribution of the sample. So sample mixture contains different proteins of different sizes. So how they get separated, how they get distributed between the two phases. So what are the two phases? Stationary phase and the mobile phase. How the different samples present in the mixture gets distributed or gets partitioned, means distributed or separated. Okay, between the two phases. That is very, very important. I told you, no, the smaller molecules are separated or partitioned onto the gels of the stationary phase, whereas the larger molecules are partitioned or present in the mobile phase. That is how they get separated. Okay, so because there is no affinity, only because of the exclusion or the filtration property. Okay, this is how they get separated. So for two immiscible phases, the mobile and the stationary phase, so here the phases are immiscible, that are, they are not soluble with each other. That is the mobile phase and the stationary phase. So you have a distribution coefficient, kd. Okay, kd value, kd is the distribution coefficient. It is a constant at a given temperature for any compound, any protein. So how is it calculated? The distribution coefficient of a compound to be separated is calculated by the dividing the molar concentration of the compound in the stationary phase by the same concentration of the same compound in the mobile phase. Suppose the compound in the mixture contains A, B, C. Okay, so concentration of A in the stationary phase divided by concentration of A in the mobile phase. So for smaller molecules, the molecules will be present more in A. It will not, concentration will be present more in stationary phase than the mobile phase. Okay, so in that case, the concentration of the sample A will be higher on the numerator and lower on the mobile phase. So the value will be 1 or more than 1. Okay, so if the analyte or the protein B is present, larger molecule B, okay, larger sized molecule B will not be present or will not be taken up by the stationary phase. So they will be present in the mobile phase. So concentration of this particular molecule B will be more in the denominator than the numerator. Okay, when you divide, the concentration of the larger molecule B will be present more in the mobile phase, that is near denominator, than the stationary phase, that is the numerator. So when you divide a smaller number by a larger number, you will get a value lesser than 1. Okay, lesser than 1. Now when the molecule is C is in between A and B. Okay, the size is not too small, not too large, it will be in between A and B. So the distribution will be somewhat different. Some will be in stationary phase also, some will be present in mobile phase also. So their kd value will be in between 0, that is 1 and lesser than 0. Okay, 1 and 0. So that is the value. Okay, so this is the kd value. So what is the distribution coefficient? Hope you have understood. It is the concentration of a sample A or sample B or sample C in the stationary phase divided by the concentration of the same sample A or B or C in the mobile phase. Okay, if it is 1 or more than 1, then the concentration of the sample is present more in the stationary phase, numerator. Okay, if it is present more in the mobile phase, then the kd value will be lesser than 1. Okay, so the average distribution coefficient will be molecules absorbed in the stationary phase by molecules absorbed in the mobile phase. So this is what I told you. Void volume. What is the void volume? It is the volume of the mobile phase present in the column in between the stationary phase. Okay, that is it is not the volume of the mobile phase present inside the gel. It is the volume of the mobile phase present outside the gel. That means in between the gels or the intermolecular spaces of the gels. So it is the volume of the mobile phase Vm. It is called as Vm or V0 in a column. Okay, in an ideal case, it is equal to the mobile phase hold up volume. For example, if the stationary phase occupies 40% of the total column, the void volume will be 60%. So the volume of the mobile phase present inside the gel plus the volume of the mobile phase present outside the gel is the total volume of the mobile phase. It is called as the bed volume or the total volume. Okay, so the volume of the mobile phase which is present outside the gel is called as the void volume. Okay, if the void volume is 60%, the volume of the mobile phase present inside the gel will be 40%. So molecules that do not enter the matrix are eluted in volume, void volume, as they directly pass through the column as the same speed of buffer. So this is the principle involved. So there is a small formula, okay, for the that is the elution volume. VE is the elution volume. So you have a formula, V is equal to V0 plus KD into VI. So VE, you know, it is the elution volume, that is the volume of the mobile phase required to elute the particular sample. Okay, the volume at which the molecule of interest elutes. Okay, the volume of mobile phase that is required to elute a particular sample, okay, in a mixture. So that is equal to VO. VO is the void volume. You can see VO is the void volume plus KD is the distribution coefficient and VI is the volume of the mobile phase inside the bead. Okay, VT is the total volume. So this is the formula. So KD can be calculated by reverse multiplication. VE minus VO by VI. Okay, so you get a formula like this: V minus VO into, sorry, divided by VT minus VO. Since VT is equal to time, okay, that is time taken, sorry, total volume is equal to VO minus VI. So what we have told, no, void volume minus the volume inside the gel. So this is a calculation to find out the distribution coefficient as well as the elution volume of a particular sample molecule in a mixture. So as I told you, if the KD value is equal to 0, so the values can be range in between 0 to 1. So when can be 1? When the value can be 1? When the numerator is higher, it can be 1. Higher than the denominator, it can be 1. Okay, when what is the numerator? Numerator is the stationary phase. Denominator is the mobile phase. So when the concentration of the molecule is more in the stationary phase, then the value will be more 1. Okay, 1 or more than 1. When the concentration in the stationary phase is lesser than the concentration in the mobile phase, then definitely the volume or the value of KD will be lesser than 1. Okay, so you can get a value 0, KD value 0, when the analyte is large. That means there is it has not entered, the larger sized molecule has not entered the gel at all. Okay, the concentration in the stationary phase or the gel is 0. In that case, it will be 0 by whatever may be the concentration in the void volume in the mobile phase. Then the value of KD will be 0. That means the analyte is too large to enter the gel, or the molecule has no interaction with the resin and passes through the column along with the mobile phase thoroughly without any hindrance, okay, along with the mobile phase. Okay, so in that case, VE is equal to V volume. So elution volume is the void volume. Okay, what is the elution volume? That is the volume of the mobile phase present in outside the gel. Okay, that is the elution volume. Now, if KD is equal to 1, so when KD will be 1, when all the molecules in the mixture that are to be separated enters the stationary phase or the gel, then the concentration of this particular molecule is more in the stationary phase and none in the mobile phase. Okay, none in the mobile phase, or the value can be in between, when some of the concentration may also be present in the mobile phase. Okay, so if the molecule is so small that it has full entry to the pores of the bead, then in that case the elution volume will be, that is the volume present in the column plus volume present inside the gel. Okay, that will be the total volume. So the molecule, when will the molecule come out of the gel? Okay, when you increase the mobile phase, or when you change the buffer. Okay, when you use a different buffer, then it will come out of the gel. Okay, just like what we have seen in the previous case in ion exchange chromatography, the molecules which are strongly attached to the stationary phase will be eluted by changing the concentration or pH or alter the molarity of the mobile phase. But here you need not alter the anything, any concentration or pH or anything, or do not use any harsh or strong chemicals. You just have to change the buffer. Okay, when you change the buffer, automatically it will come out of the gel and will be eluted in the end. So due to the variation in the pore size between individual gel particles, there is some inner mobile phase which are not available and some available to analytes of intermediate size. Okay, some because of the pore size varies and the pore will allow entry of only smaller molecules. Some other molecules no, are present of intermediate size which may just enter or which may not be able to enter and which is not as large as the largest molecule. So their KD values will be between either between 0 to 1. Okay, so for analytes, two different relative molecular mass and KD values. Okay, these are the different KD values. So for that, the elution volume will be this formula. Okay, K dash D minus K double dash D into Vi. So this is how the KD distribution coefficient you have to understand. It is the divide. It is got by dividing the concentration of the sample, that particular sample, in stationary phase divided by the concentration of the same sample. That is very important in the mobile phase. Okay. So here you can see the VO, that is the void volume, the volume of the mobile phase present inside the column but not inside the gel. VE is the volume of the mobile phase present inside the gel. Okay, and VT is the total volume which is present totally in the column, that is both the inside the gel and the outside of the gel. Okay, so this is for very small molecules that have full access to the pores of the bead. Okay, small dots. Okay, bigger ones if they will be present in the void volume, smaller ones will be present in the this one. Okay, inside the gel. Okay, now this is the principle how the thing. So as I told you, this is the gel. Okay, so gel is made up of like a ball shaped structure which will be a dot or some powder. When they are dissolved or immersed in a suitable solvent, what will happen? The gel will swell. How do they swell? They have pores. You can see many pores here. Okay, white color pores. You can see how the molecules enter the gel. So this through the pores, the mobile phase or the buffer, okay, the buffer will enter the gel. So when they enter, the gel will swell. Okay, now you can see the proteins of different sizes. Yellow colored ones are bigger and red colored ones are smaller. Okay, so those smaller sized protein molecules, they can just enter the cells. So the molecules which are smaller than this also can enter the gel. Okay, so what happens with the molecules very smaller than this can enter the gel and they come out of the gel also. Okay, they can easily come out of the gel. But here these molecules which are perfectly the size of the pores of the gel get entrapped in the gel. Okay, so the molecules bigger in size cannot enter. The molecules much smaller in size than the red colored ones will enter and also come out, can easily come out. But those molecules which are just the same, the diameter is the same as the diameter of the pore, when after they enter they get entrapped in the gel. So the size of the molecule vary. They may be big like the yellow one, they may be exactly the same diameter as the pore size of the gel, or they may be smaller than the pore size of the gel. Okay, the ones which are smaller than the pore size will come out of the gel very easily. They'll enter, pass through it, and they may come out. Okay, or the bigger ones may not be able to enter at all. But those ones which are appropriate and whose diameter is exactly the same as the gel. Okay, gel is a soft structure, yeah. So they get, once they enter, they get entrapped. Okay, like this. So those bigger molecules which do not which are not entering the gel, they flow out of the column first. Okay, they can be desalted proteins. So it's here one application is desalting of the proteins. So the proteins can be removed from the salts. The bigger proteins are collected first and the salts are collected as molecules and entrapped in the gel. Thus they can be separated from the salt. The protein can be purified from the salt. Okay, that is called as desalting. So here you can see the thing, molecular column, exclusion column, how the mobile phase is passing through the column, and you can see the gels. See, these are the gels. So this is showing the different pores, and you can see the volume inside the that is in between. I told you know the space in between the gels, that is called as the void volume. There is a space inside the gel, that is called as the elution. That is the volume inside the gel, Vi and Vo. Okay, the total volume of these column is Vt, that is the volume present inside the gel and outside the gel, and Ve is the elution volume required to elute a particular sample. So here you can see three different sized samples are allowed to pass through the column. So one is the large molecule. See, these molecules do not enter the gel, so just like that, they easily pass through the intermolecular spaces and come out first. Okay, the second one is green sized molecule. They are intermediate molecules which are not as big as yellow and not as small as red. So they cannot, they are partially trapped and partially not trapped. So what happens? They will come out, they will try to enter, but they will not be able to enter. So they will come out second. Okay, the next one, the third one is red colored, or you can say pink colored ones, which can easily, so here also not all the samples are entered, some are present in the outside also. Okay, so mostly majority are trapped inside the gel, so they get trapped inside the gel, and after all the green colored ones and the yellow colored ones are washed out, okay, washed out, finally they will be brought out of the gel by adding a different buffer. By changing the buffer, they will be eluted out of the gel. So the proteins get separated one by one, first yellow, second green, and the third one pink colored ones. Okay, at the appropriate time interval. So a mixture of molecule dissolved in liquid, that is a mobile phase, is applied to the chromatography column, this is the procedure. Okay, it is applied to the column which contains a solid support in the form of microscopic spheres or beads. So as I told you, the mass of beads within the column is often referred to as the column bed. The stationary phase is called as the column bed. So you can hear these words: column bed, stationary phase, total volume, void volume, elution volume. இதெல்லாம் வந்து are the terms which are given in the chromatography. So the beads act as traps. So I told you, the entrapment, or they act as sieves or filter and function to filter small molecules which become temporarily trapped within the gel. So first, which molecules are excluded first? Larger molecules are excluded from the beads first. So large sample molecules cannot or can only partially penetrate the pores, whereas smaller molecules can access most or the all pores. So these are medium-sized molecules which may not be either fully able to enter, okay, enter the pores. So the larger molecules elute first, smaller molecules elute later, while molecules that can access all the pores elute last from the column. Those molecules which can enter complete access into the gel bead, smallest ones will be eluted last. So particles are separated depending upon the size. So what is the media used? Stationary phase. So stationary phase should have a lot of properties. Okay, it should have a lot of properties, as I told you. Here it should swell. Okay, and the size, it should have some cross-linking polymers. It should be made up of some molecule which are having cross-linking polymers, which will give the pores. Okay, which will give the pores. So it should be semi-permeable. It should be porous, and the size of the pore should be well defined. Only then you can select what molecular size proteins can be separated by what kind of gel. Okay, so the molecular weight of the protein can be measured in terms of Daltons. Okay, so accordingly the pore size also can be defined, well defined, by increasing the concentration or differing the cross-linking. So beads are cross-linked poly only when they are cross-linked. Just like as I told you, just imagine the cross-linking between agarose and this one, amylose and amylopectin. There are so many cross-linking which we will come across. Okay, so degree of cross-linking on that will only control the pore size. The smaller pore size is used for desalting, purification of the proteins from the salt. Intermediate size proteins are used to separate relatively small molecules. Okay. Very large pore size used for that is big biological protein molecules, complex molecules. Now, what is the stationary phase used here in gel filtration chromatography? It is called as dextran. So we have seen the previous class. Dextran is a carbohydrate. So example for a very good dextran is Cefadex. Okay, Cefadex. This is one kind of stationary phase used. What is it? Dextran. Example is Cefadex. Okay, it is a carbohydrate. Then you have acrylamide. Okay, polyacrylamide. This word you would have heard very much. Polyacrylamide. Okay, acrylamide is a compound used in SDS page. Okay, so acrylamide, polyacrylamide and disacrylamide. These two combine together to form cross-linking. So acrylamides will monomers of acrylamide will link together to form a horizontal chain. Okay, bisacrylamide is two molecules of acrylamide joined together by a methylene bridge. So that will act as a cross-linking bridge between two horizontal polymers of acrylamide. Okay, and also you have dextran polyacrylamide. This I will show you the structure of polyacrylamide when we speak about SDS page. Example is cefacryl. So cefadex and cefacryl are the very good commercially available stationary phases used for gel filtration chromatography. Okay, so they are available in variety of sizes, variety of pore size, okay, which will enable the separation of different molecular weight proteins. So by selecting these kind of gels, you can determine the molecular weight of the proteins that are being separated. Okay, so what is the properties of good stationary phase? Enna irukano? Okay, it should be chemically inert. That means it should not have any reaction with the sample. It should also be inexpensive. அப்பதான் எல்லாராலையும் ஈஸியா யூஸ் பண்ணி செப்பரேட் பண்ண முடியும். It should not be very expensive. It should not react with component to be separated. That is what it should. If it is inert, it should not react with the sample. It should not react with the element. Even with the mobile phase also, it should not react. It should be colorless, uniform in size and shape, and mechanically stable. ஸோ ஏதாவது வேற ஏதாவது பஃபர் ஆட் பண்ணால் அது மாறக்கூடாது, மெக்கானிக்கல் ப்ராப்பர்ட்டிஸ் சேஞ்ச் ஆகக்கூடாது. Now, the gels can be classified into three different types. It can be soft, it can be semi-rigid, or it can be highly rigid. So, highly rigid இருந்தா ஒஞ்சு போயிடும். Okay, so soft na ரொம்ப soft. Example, cefadex. Cefadex is a type of dextran. Okay, carbohydrate. Polyacrylamide gels. இதெல்லாம் வந்து two kinds of soft gels that can be used for the separation of proteins. So semi-rigid gel oda example enna biobeads. Okay, some commercially available biobeads are there. Okay, so what is it used for? Separation of non-polar polymers in non-polar solvent. So non-polar polymer separation they are used. Then highly rigid gels. So they are wadaira madri illaya glasses. So they are used for the separation of polar systems. So these are the three kinds of gels used for gel filtration chromatography. So mobile phase eppadi irukano? The liquid used to dissolve the biomolecules to make the mobile phase is called a buffer. So இங்க என்ன சொல்றோம், mobile phase usually we call it as a buffer. So buffer னு கூப்பிடுறோம் இங்க. Okay, so the mixture of biomolecules dissolved in the buffer is called the sample. So sample எப்பொழுதுமே before applying into the column, it should be dissolved in a buffer so that it will absorb the buffer and swell in size. Only then they can attain the property of a gel. Now the choice of the mobile phase எப்படி இருக்கணும் for separation, this should depend upon the type of separation. So என்ன மாதிரி separation யூஸ் பண்ண போறோம், so அதுக்கேற்ற மாதிரி you should choose the mobile phase. So என்னென்ன example, it can be tetrahydrofuran, chloroform, or dimethylformamide, or some other buffer. So the column, so inge mostly you can see the YouTube, mostly shorter columns are used. Okay, why? It can save time, and also the mobile phase used can be little. Okay, so small particles, that is of a chinna 5 mm. So typically 5 mm provide a better resolution. Okay, on the other hand 5 mm or even 3 mm packings are more sensitive towards contamination by sample containing impurities. Okay, so particles as large as 20 mm, so this can be used for separating 5 mm particle, and 20 mm have been recommended for high molecular weight polymers. Okay, அதே மாதிரி columns with different porosity or mixed bed columns also available, which will provide a better separation. Okay, so usually சின்ன column size தான் யூஸ் பண்ணுவாங்க. Okay, 5 mm or 20 mm columns. So what are the advantages? Proteins can be separated at a faster time. Time taken is very slow, very less. Well-defined separation. Nalla paakla. If I'm going to show you a particular YouTube where you can see how well the separation has taken place without any contamination. And the bands are narrow with good sensitivity. Sample loss kadayadu. Okay, small amount of mobile phase are required. No large volumes of mobile phase may be required. The flow rate can be set. We can adjust the flow rate. So disadvantage is in a limited number of peaks that can be resolved within the short time scale. நிறைய sample இருந்தா பண்ண முடியாது. Filtration must be performed before using the instrument to prevent dust. So எப்பொழுதுமே pre-filters should be used, okay, to avoid any contamination of the mobile phase into the column. Okay, so that the the contaminants may not interfere with the sample. So when the detection வரும்போது இது sample தானா இல்ல வேற ஏதாவது contaminant அப்படின்றது confusion இருக்கக்கூடாது. So application, it can be used to separate proteins. So very important application is separation of proteins. Okay, ion exchange la kuda amino acid separate பண்ணலாம், protein கூட separate, protein hydrolysate கூட separate பண்ணலாம். ஆனா இங்க வந்து mainly used for the separation of proteins. Bigger molecular weight proteins also can be separated. So the enzymes, hormones, nucleic acids, and even viruses can be purified by this kind of gel filtration chromatography. And you can determine the molecular weight. ஓகே, குளோபுலர் ப்ரோட்டீனாக இருந்ததுன்னா, குளோபுலர் எப்படி இருக்கும், ரவுண்டாக இருக்கும். ஃபைப்ரஸ் வந்து ரிபன் ஷேப்டாக இருக்கும். ஸோ தோஸ் ப்ரோட்டீன்ஸோட மாலிகுலர் வெயிட்டை கண்டுபிடிக்கலாம். Then you can also use this particular gel filtration chromatography for the separation of sugar, proteins, peptides, rubbers, okay, based upon the size. Okay, so it can also use to determine the quaternary structure of purified proteins. So quaternary structure na, when more than one polypeptide chain is involved in the protein structure, this can be used to separate them and determine the quaternary structure. Okay, it is widely used technique for the purification of and analysis of synthetic and biological polymers such as protein, polysaccharide, and nucleic acid. Then the RNA virus also can be purified using, instead of Cepadex, agarose gel can be used for separation. So stationary phase மட்டும் மாத்திட்டா we can use different stationary phase for the separation of not only proteins, sugars, nucleic acids, RNA, and viruses also can be separated. A very good technique for desalting of the proteins. Okay, so this is these are the thing you can reference you can use. I am sending you Wilson and Walker, you already will be having. Okay, and I'll send you the PDF of gel filtration principles and methods by Amershan Biosciences. You can refer them. Okay, now you can watch a video of gel filtration chromatography.